Well, good morning, everyone. And on behalf of James Polly and myself and Visha Krishnan and FDA organizers of this workshop, I would like to welcome all of you to this Shady Grove campus for our third in-person workshop on mitigation strategies for nitrosamine drug substance-related impurities, quality and bioequivalence considerations for generics. So maybe next slide. It used to work before much, but okay, that's okay. I'm always the first one. So I think it's really great to see you all in person. It's our third in-person workshop. You probably know about the center. We were established in 2020 during pandemic because Jim and I received this grant from the FDA, U19 grant, to organize this Center for Research in Complex Generics. We have a mission to increase access to safe and effective generic drugs through infrastructure, communication, education, and research collaborations across both industry, as the center, and academia and FDA. So we are dedicated to advancing the programs to stimulate scientific dialogue, disseminate current insights, generate new knowledge about complex products. And we've been around for now 2.5 years. I think the best compliment I received was in the GADUFA public forum and just a couple months ago when somebody in generic industry called us an honest broker. So we are an honest broker of information between industry and the agency. We have three main missions. The one is to this infrastructure and communication, and through this mission we interact with many generic companies as well as other stakeholders to understand your main challenges that you're experiencing. And clearly nitrosamines came up very loud and clear in 2000, almost at the start of the center. The other mission of us is education and training. So organizing workshops like this one is one of the core functions of the center. And then we are moving toward collaborative research, first with the small projects at both universities, but going forward, we would like to be intermediary between industry and the agency and try to encourage some joint research between multiple companies. And to me, nitrosamines is one of those examples when you guys, innovators, generics, all come together to form consortium to solve the issue. So it's absolutely possible when we pick up a scientific issue for all of us, work together, and I hope we could be a great intermediary for that. In this interview process, we talked to many companies, many generic companies and some stakeholders, some makers of software, some CROs. So some of them are here on the list. If any of you here in the audience, but also online, have not interacted with us, please reach out to us. We have website and we have some contact information at the end of those slides. We usually just use this as a great opportunity to get the information and then be disseminated in an unbiased manner. And we also use this information to design which workshops are important, which research is important. So it's good for us to have a right information from the ground. So nitrosamines is clearly a growing concern. This, maybe not growing, but a very large concern. It came up in 2020 during COVID-19 pandemic. Somebody told me that if it wouldn't be for COVID-19 pandemic, we would be talking about nitrosamines all the time. The major comments initially was that the original guidance had very no separation between simple nitrosamines and complex nitrosamines, was focused on more simple nitrosamines, and the limits are very strict. So many companies brought up issues such as, many of our drugs we are using only for 1 week, like a course of antibiotic treatments, but at the same time, the limits we have to set up are the same as it would be dosed chronically for for years, like statin. And even statins, they're not prescribed from the early childhood. They usually come and play when you are in 50s. So how do you generate these limits? That's a very important consideration. There was no general lack of analytical methods and clarity in method development and those limits and methods to generate risk assessment. Eventually, there was a clear refocus from simple nitrosamines to more complex nitrosamines, but there is not enough experts in analyzing complex nitrosamines. Every complex nitrosamines require reference standards, its own analytical method, and this is how the companies start forming consortiums to focus on specific complex nitrosamines. And then this whole lifetime exposure that I mentioned before. Then how do you mitigate the strategies? How do you limit those nitrosamines in the product? Companies found out that they see nitrosamines not only in the drugs themselves, but in many excipients that have some reactive species. And most common excipients like carboxymethyl cellulose and others have those substances that would react with the drug and generate nitrosamines. It was not clear when they are made. Are they in the raw material, in API? Are they made during process? Are they made during storage? And then also how best to analyze them? Are they made during analysis? How do you validate analytical method? There were some strategies to mitigate it, and the agency came up with some of them: use of antioxidants, change in pH, and we will see some other great presentations today. But then the industry is concerned how would it impact manufacturability, stability, and the agency is somewhat concerned how would it impact absorption and bioavailability. So in general, it is a big topic. We are all getting better. There is a lot of experts today to discuss all of this, and hopefully we'll make a good progress today and over the next years. But for generic industry, it's especially important topic because you produce so many products and many products are affected, and it just puts a very large burden on analytical development. And it's important for us to have access to those drugs. As a center in our education mission, today we at nitrosamines workshops, we have a couple more planned for this year. One is in October, advances in PBPK modeling and regulatory utility for oral drug product. Then in December, another very important for complex generics is characterization of complex excipients and formulations. And then we are moving to next year, another comparative user interface for drug device combinations going toward next year. We'll let you know when it will be announced. We have some small research projects, some are around specific complex injectables, some are basic around orally absorbed drugs and how best to measure dissolution and correlate with absorption, some around inhalation product and PLGA. There were some of the research projects completed. They're all small volume projects. Jim Polly and I run the center. He is at University of Maryland. I'm at University of Michigan. Everybody know Visha Krishnan. She really runs the center. She is at University of Michigan. Dana Hamill is with us in person. She is great in coordinating of all of those workshops. And Jen Dick is admin for the center. She is based in Michigan. Here are all of our contact information. We have a website, we have YouTube channel where we post all the presentations. We have presence in LinkedIn and on Twitter. Many people join us on LinkedIn, and that's a good way to know about upcoming events. Now Visha will come in Gadufa Public Forum. We said that we will describe our new website. So we reorganized website, making it very searchable. You guys could find the outcome of all the Gadufa research, not only papers, but also abstracts. So she will speak about it very quickly.
Hi, everyone. Good morning. Thank you all for being here in person, and thank you to all those who have signed in online. Just for numbers, we have over 2,500 people who've signed up for virtual attendance. And we have a really nice group here in person, too, for those who are online and are not able to see the group here. As Anna mentioned, we are developing a new website, and the whole goal of that website is for it to be a good resource for people who are developing complex generics products. That will feature, I'll wait for Jeff to play the video, and then so that's our homepage. It has information on events, upcoming events, and the key, and it will house all of the resources from our events. And as Anna mentioned, the GDUFA research outcomes, which is a big part of the website. These GDUFA research outcomes would include posters, presentations, any publications that came out of GDUFA-funded research. The database will be searchable with keywords, and you're able to sort and find posters, presentations, and you're able to download these resources as well. And that's what distinguishes it from currently accessible resources. Another aspect is if you are not looking for a particular website, but you just want to browse what's there, you're able to browse these research outcomes too, which are categorized into 9 different categories. And if you were to click on any one category, it'll tell you what are all the research publications, posters, presentations that are available. And all of these can still be downloaded. Wherever there is a PDF, there is a PDF preview option, and you're able to download it, share it with people, share it on social media. This is something we're excited about because this will be a very valuable resource to the community that's developing complex generic products. Another aspect of our website, a key aspect, which a lot of people email us about, is how do I find presentations and videos from our events? So we will have, our current website already has this information. The new website will have it organized in a more user-friendly fashion, where you're able to find information about the event. You can look at speaker bios, the event outline, and I'll let it go to the speaker bios, so you're able to see the speakers. As you noted, in most of our events, we don't spend too much time on the bios because we want to move on to the content. And we will have all of the videos posted where you can play it on our website or link it to YouTube. We will also have slides that are downloadable, very similar to what you saw for the GDUFA research outcomes. There is a PDF preview option and a download option for these. The last element of the website, which is something which we hope a lot of people will start using, is collaborating, which will come up in a second, collaborating with us. If there is interest in collaborating on research opportunities, or if you want to be a speaker or a panelist at any of our upcoming events, we tend to announce these events ahead of time. So 2024 events would be announced very soon, and then by end of summer, so you're able to reach out to us and tell us if you're interested in being a speaker or if you want to collaborate with us on research opportunities. This website is planned to be launched by end of summer, and there will be a notification that will be sent out to our entire listserv. And I also want to note that for those who are looking at all the content on the website, this is just a snapshot, and any of the content there is just placeholder content. So if you notice something about upcoming events and you're wondering if that's an actual upcoming event, it's just a placeholder for us to just showcase the website. With that, I will like to introduce Dr. Robert Lionberger to provide the opening remarks. Dr. Lionberger is the director of ORS at OGD, following which we will have Dr. Andre Ra provide the workshop overview. Dr. Andre Ra is the Associate Director for Science and Communications at OLDP. Thank you.
Thank you, Visha. So it's very great to be here today with all of you and see people here in person as well as the thousands of people who are online. It shows the great interest that there is in this topic. And first, I want to thank and recognize the Center for Complex Genera under leadership of Anna and Jim for working on this. And I want to explain a little bit why that was sort of exceptional. Again, this is the Center for Complex Generics. Again, the focus of this center when we developed it was to really facilitate the development of complex generics, because that's where we thought the key scientific and regulatory challenge facing the generic industry were. But I think through the interviews that Anna and Jim conducted across the generic industry with many of the stakeholders, kept hearing constantly again that the biggest scientific challenge facing the generic industry was related to nitrosamines and what do we do about this and how do we get this under control. And I really want to thank them and recognize them for being able to be flexible and adaptive in order to, something that may be a little bit out of scope of the original intention of the center, but still very critical to the generic industry. And I think that's a fantastic element of this. And as we move forward in this area, something that adaptability and flexibility is something both from FDA and from the generic industry that you'll have to do in order to right the ship, renormalize our system after we deal with this. And so I think it's a really great example, and I really appreciate them specifically for adapting to that, participating in this, engaging in this topic, and working with FDA and our coordinating team on that to get something on point for this. And for this workshop here, again, although the nitrosamine-related impurities certainly affect they're found in brand and generic products, the focus of this workshop really is on the impact on the generic industry, because there's some specific and unique challenges there that we want to make sure are recognized and are discussed. And they really come from the fact that, in almost all cases, drug substance-related impurities are things that are handled during drug development, where the brand companies that are developing new molecules have infrastructure around that, where FDA, as we're reviewing new drug applications, IND applications, all of our pharm tox reviewers are linked in with the new drug review division, so that the main focus of our evaluations of drug substance related impurity generally happens during new drug development. But when we find something new later, that could be a challenge to the system, right? The generic industry isn't set up to, all right, I have to understand and qualify these new impurities. Our reviewers in the generic drug side generally are there, that's not the focus of our pharm tox group as well. And if one of these happens, it's something that can be handled. But when it's a whole class, that really is a shock to the system. And again, that's why we have to be flexible and adaptable in identifying ways to efficiently move forward. And I think Anna mentioned collaboration. Certainly, we encourage the industry to find ways to collaborate. If the Center for Complex Generics can facilitate that, or there's other groups that we really support that, because we recognize this is a different challenge, right? There are perhaps 20 generic companies making the same product. Who's responsible for understanding, generating new data about a newly identified impurity, right? If everyone's responsible, sometimes it ends up no one's responsible. And I think that's the challenge here. And I think, this workshop and many other workshops that FDA is engaged in across, the global community are parts of that. So I encourage people to be looking for that collaborative aspect, recognizing that this is sort of a very specific and challenging and unique situation. But I think that's why these events that the Center for Complex Generics organizes are so important. So one of the hallmarks from the generic drug program, our communication strategy, is we have workshops where it's FDA speakers talking and communicating to industry like what we're doing, what does our guidance mean, how to do it. But for the workshops hosted by the Center for Complex Generics, we intend them to be different than that. We have lots of speakers from industry, from different experts, from related suppliers to the industry, experts in specific fields that support the industry. And we also have FDA speakers, and we have panels that include people from across those different groups. I think this is a really important place to have those interactions and really understand what are the unique challenges that face the generic industry as they try to deal with these drug substance related impurities. And I think that can be very helpful to us at FDA to understand the constraints, the capabilities of the different components of the industry. And I think that will help us develop better and more flexible and adaptable approaches to this as we move forward. So that interaction is key to what we're doing today. And so I really look forward today. We'll start off really with a discussion in the morning about, where do these impurities come from? What are the mechanisms to mitigate them? We'll have a session, update on what some of the current thinking on the sort of safety evaluations of these are. And then, to me, one of the most important sessions is the final session where we talk about the potential, implementation of mitigation and its effect on bioequivalence and reformulation. And the challenge here, right, is that, it seems like, the first session you'll hear that there are things that you can do to reduce nitrosamines in drug products, but many of them involve reformulations. And if you look at our older guidances on post-approval changes, they'll say, well, if you add an antioxidant at a certain level, right, these become what we call level 3 changes, new bioequivalence studies. That can be a very serious and challenge to do this. And, when we look at the scope of this, how do we all want less nitrosamines in drug products. And so we want to do whatever we can to facilitate the efficient reformulation. So you'll hear in that session about the research that we've done to try to identify the factors that may affect, the factors of the potential reformulations that may impact bioequivalence and how significant they are. And that's very important to a science-based approach for that. And again, as you're moving forward on this, we encourage you to interact with the FDA through. There's a lot of new mechanisms in GDUFA 3 for the generic industry, post-approval control correspondence, post-CR control correspondence is where you have specific questions. You're considering a reformulation, you want to interact with us and get feedback on what data can support that. We'll talk about the science today. You can interact with us specifically about your application through those meeting and control correspondence mechanisms. Again, you'll hear a little bit of updates on guidances. So definitely, we'll be writing guidances on this that will hopefully make it even more clear how to move forward. But again, really appreciate the Center for Complex Generics work and all of you being here to really help develop flexible and adaptable approaches that will help the generic industry move through the situation and deal with these effectively and reduce the amount of these impurities in the drug products that reach the American public. So thank you all for being here and look forward to engagement all day. Thank you all very much and welcome Andre to give his perspectives on this.
Yeah, thank you, Ralph, for the introduction. So I'm going to start off, with this workshop again. My name is Andre Ra. I think, Robert introduced me already. Uh-oh, how do I get this out? Okay. Okay. I'm the Associate Director for Science Communications in the Office of Lifecycle Drug Products in the Office of Pharmaceutical Quality. And then specifically, I would like to thank the Center for Research on Complex Generics for this opportunity to host this very critical workshop on the mitigation strategies for nitrosamine-related impurities. And it's also as Rob indicated, the potential bioequivalence implications of those mitigations approaches. So, since I'm from the Office of Pharmaceutical Quality, we always start off with a definition of what quality is. And a quality product of any kind consistently meets the expectations of the user, and drugs are no different. Patients expect safe and effective medicine with every dose they take, and pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects, and it is what gives patients confidence in their next dose of medicine. Okay? So, as background, I just want to give a very, very short, brief background of nitrosamines and the various classes. So nitrosamines describes a class of compounds having the chemical structure of a nitrosyl group bonded to an amine, okay? And that's shown in that slide here. And one of the principal, but I wouldn't say, I would say this is the principal mechanism, but I would not say this is the all-inclusive mechanism. So I want to be very clear about that. But probably the most predominant mechanism by which they form is by nitrosating reaction between an amine and nitrous acid, or it could be nitrite salts under acidic conditions. And really the reason we're really worried about these is that nitrosamines are potent genotoxic agents in several animal species and are classified as probable and possible human carcinogens by the International Agency for Research on Cancer. and they are referred to the cohort of concern compounds by ICH M7. And really, there are really two classes of nitrosamines, that have a broad impact. It's not only the generic industry; they're also having a broad impact on the innovator industry as well. And we have some people from the innovator industry even speaking at this symposium. It's such a broad impact. So the first class is the so-called small molecule nitrosamines, such as N-nitrosodimethylamine, and nitrosodiethylamine, and so forth. And these small molecule nitrosamines constituted sort of the first wave that impacted the pharmaceuticals, dating back to 2018 when we first saw them in valsartan and later on in other pharmaceuticals. And these small molecule nitrosamines are what their name implies. They are low molecular weight nitrosamines that, for the most part, don't share structural similarity to the active ingredient. And generally speaking, these small molecules, this is generally speaking not always the case. There are some exceptions. They're formed during the synthesis of the API, and they arise due to the low levels of secondary amines that may be present in avid solvents such as DMF, or tertiary and quaternary amines that are commonly used in synthesis of these APIs. And the presence of these residual amines during the synthesis of the APIs in conjunction with residual nitrites used in synthetic steps, such as in the quenching of azides, is what contributes to the formation of these small molecule nitrosamines. And these small molecule nitrosamines are very potent carcinogens. We do know that in animal species, and because of that, they need to be controlled at very stringent levels around 26.5 nanograms per day. But there's one thing that, I'm not saying it's a good thing, but these oftentimes, because they're formed in synthesis, we can generally control them. API suppliers can alter their synthetic pathways to avoid these, nitrite quenching reactions. They can avoid these, DMF solvents, and they can also add additional purification steps to ensure that these small molecule nitrosamines can be effectively controlled to these very, very low levels, so they don't present an exposure risk to patients. But really, the reason we're here today is not to talk about small molecule nitrosamines. We're here to talk about the second wave nitrosamines that we're seeing in pharmaceuticals. And these are the so-called nitrosamine drug substance-related impurities. And really, these are different, from the so-called small molecule nitrosamines, in that these nitrosamines share structural similarity to the active ingredient or a fragment of that active ingredient. And given they have structural similarity to the API or a fragment thereof of that API, they are also structurally quite diverse. And there are many more of them than there are the small molecule ones. So the next slide, gives the primary root cause of, the formation of these NDSRIs, okay? And again, I want us to be very clear. This is not the only mechanism by which they form. There are other mechanisms, but I would say to you, this is probably the predominant mechanism by which they form. And the root cause is pretty, conceptually simple. Many active pharmaceutical ingredients contain secondary amine functionality, a good chunk of them in our drug, substance repertoire. And these secondary amines are susceptible to the reaction with residual nitrites that are ubiquitously present in drug product formulations. They could be present in the solvents, or they could be, and particularly we know that we're going to have lots of talks showing that they're present in excipients, and they can come, air, can contribute to the formation in excipients. There'll be some very nice talks showing that. And that simple combination of a secondary amine, either from the API itself, or even a fragment of that API with nitrites, leads to the formation based upon when these well-known principles to the NDSRIs in the drug product, either during the product's manufacture or shelf life. But there's one big contrast, to the small molecule nitrosamines. Actually, there are two big contrasts. The first is that, in small molecules, you could see them in synthesis, and you could, develop steps to purge them, to get rid of them quite effectively. But NDSRIs are often generated in the drug product. So if the conditions are favorable in the formulation to the formation, and once they're generated, you're stuck with them in the drug product. You can't just clean up the drug product as you could have cleaned up the API. And also the other challenge that we have with these nitrosamines is, the small molecule nitrosamines, we know what the safety limits are. We can define what the acceptable intake levels are. But the NDSRIs are sort of a class of nitrosamines where their immunogenicity, carcinogenicity is not well completely understood. And that really presents challenges to defining the acceptable intake levels. So as you can see, nitrosamines is the nitrosamine drug substance related impurities is quite a complex challenge. And because of this, it requires really a multidisciplinary approach to find an eventual solution to this problem. And we will certainly find that. And because of that, we divided this workshop conceptually into 3 parts, okay? So in the first session, we will discuss the various risk factors that contribute to these NDSRIs, whether it's from APIs or from excipients that may contribute to the formation of NDSRIs in drug products. And we'll also discuss the analytical methodology that can be used to, quantify these unique nitrosamines. But but the good thing is that we do know that there are strategies to prevent these NDSRIs in the drug product. This could be employed by either controlling the nitrite in excipients or by judiciously selecting the excipients during your initial formulation development. There'll be a lot more discussion on this on the use of inhibitors or scavengers such as antioxidants or pH modifiers. And there'll be some presentations showing they can very effectively, mitigate the formation of these NDSRIs. So that'll be the first session. So in terms of the second session, as I mentioned, unlike small molecule nitrosamines, NDSRIs, the carcinogenic potential is not very well understood, okay? So while we know that the nitrosamines are a cohort of concern, and some are definitely carcinogenic, not all nitrosamines are carcinogenic. So there's going to be a discussion in the second section on how FDA and drug product manufacturers can assess the potential risk of NDSRIs in human health by using testing approaches, that are being developed. There's going to be a talk on, for example, optimizing the aims to be more relevant, and there are going to be other talks as well. And, there also is going to be a discussion on, predicting their potency of based upon quantitative structure activity relationships or QSAR modeling or other relevant tools. And with this, we hope to be able to establish clinically relevant intake levels based upon these unique NDSRIs tailored to its unique BMH. NDSRI is different. And so we should hopefully, with time, develop, methods to tailor acceptable intake levels, particular to their mutagenic or carcinogenicity profile. And then finally, as I previously mentioned, and Rob already alluded to, we will have, we're already going to have 2 talks that are going to talk about the use of inhibitors or scavengers, such as antioxidants, to mitigate the formation of NDSRIs. These are going to be presented in session 1. So in session 3, we will discuss the impact, of such reformulations that employ these approaches. And I think what Rob was alluding to, yes, if you do these reformulations, based upon the SUPAC guidances, yeah, you're going to have to do an in vivo, PK study. But I think what we're going to discuss, and this is some of the research that FDA is doing, is, can we use some of this research findings, based upon what we have, and utilize modeling simulation approaches to assess the risk of bioinequivalence should such reformulations occur. And if, if the risk of bioinequivalence is deemed to be very low, maybe we could entertain alternative approaches that may not need to employ, the PK studies based upon scientific standards. So, and finally, and last of all, I really want to thank the organizers, of course, the CRG team for making all this happen. Also, I really want to thank Dr. Kondakor Elam. He's a senior staff fellow from the Division of Quantitative Methods and Models. Okay, so I think while they're putting together, the slide, I wanted to start off with the session number 1, okay? And I wanted to, which in the session number 1 is the risk of is discussing the risk factors that contribute to formation of NDSRIs in drug products and strategies to mitigate these risks using formulation design principles. And so I wanted to sort of introduce, the speakers of this session, and then later the panel. So the first speaker is Justin Moser. He's a principal scientist from Merck, and currently his role is in actually vaccine drug development, focusing on formulation process design. But prior to that, Justin led the drug product and nitrosamine risk evaluation team, chartered with building data and understanding that can be utilized, utilizing the risk assessment of these NDSRIs at Merck. And he took over leadership of the overall cross-functional N-nitrosamine core risk team, which authored and submitted the risk assessment outcomes to health authorities. Justin Moser has a BS in chemical engineering from Pennsylvania State University and has been working, for Merck for 21 years. And the next speaker of the session will be Dr. Jingyue Yang. She's a research scientist in the Division of Pharmaceutical Analysis in the Office of Testing and Research in FDA. And in her various roles, Jan develops and applies analytical methods to assess the quality of pharmaceutical products and investigates adverse events related to the use. And she has been a key member to provide analytical support to FDA's task force responsible for investigating nitrosamine contamination in pharmaceutical products, having developed important analytical methods to detect and quantitate these various nitrosamines. Jan received her PhD in chemistry from Washington University, St. Louis, Missouri, and her bachelor's in chemistry from Beijing University in China. And the next speaker will be Sander van Gessel. Van Gessel holds a Master of Science in Chemical Engineering from Groningen University in the Netherlands and has been working for DFE Pharma, a leading excipient manufacturer. And excipients are very relevant here. As director, Sander is currently responsible for DFE Pharma's global portfolio of excipients, including lactose, MCC, super disintegrants, and starches. All excipients we know that may have nitrites. Recently has led DFE Pharma's investigations into the role of excipients and how they play a role in nitrosamine formation in drug products through nitrite and nitrate precursors in excipients, as well as numerous other projects. And the next speaker that will be Martin Erlaert, and he's the Vice President of Global API and R&D in Apotex. Dr. Martin Erlaert obtained his bachelor's in applied chemistry from McMaster University in 1987, and he obtained his PhD in chemistry from the University of British Columbia in 1992. And, in 1998, he joined Apotex Pharmaceuticals, serving in various capacities within API, R&D, and operations. And he moved to Apotex Incorporated and currently holds the role of Vice President in Global API, R&D. And the next speaker we'll have is Dia Sakleya. He's a senior research scientist within the Division of Product Quality Research within FDA. And his area of expertise include drug product quality, including opioids and regulated bioanalysis and pharmaceutical analysis. And in his current role, Dr. Sakleya leads regulatory science work related to nitrosamine impurities, including projects related to the mitigation strategies to reduce the risk of nitrosamine impurities in pharmaceutical drug products. And he'll talk about one of those mitigations approaches today. And Dia received his PhD degree in pharmaceutical sciences from Mumbai University in India and a postdoctoral fellowship from West Virginia University. And then after that, we'll have another talk on scavengers, and this will be from Dr. Marco Trumpos. He's a scientist in the early development for drug products in Sandoz Development Center in Slovenia. And Dr. Marco Trumpos works as a scientist in early development department in Sandoz Development Center in Slovenia. He obtained his master's in pharmacy degree in 2015 and a PhD in chemical sciences in 2019 at the University of Lisbona. I'm sorry, I can't pronounce it, in Slovenia. I apologize, okay? But since joining Sandoz in 2021, his main responsibilities have been the development of novel analytical approaches for nitrosamines and complex APIs, utilizing his skills in synthetic organic chemistry and various spectroscopic methods. And finally, the last speaker will be Ron Grahek. Ron Grahek is head of analytical research at the Sandoz Development Center, again in Slovenia. He received his bachelor's degree in chemistry and PhD from the University in Slovenia. Again, same, I can't pronounce it, I apologize. And so, for the past 25 years, Rock led a group of researchers as a department head dealing primarily with impurity profiles, isolation, identification, and trace analytics. And Rock has authored and co-authored nearly 40 patents and scientific papers and related to these topics. So I'm looking forward to them. So I guess the first speaker will be, Justin. So, go ahead. And I'm going to sit down there because I can't see the presentation.
Good morning and good afternoon and good evening for those overseas. Thanks. First, I'd like to thank the organizing committee for the invitation to speak today. I've been out of this topic for a little over a year now, so it's great to refresh and reconnect on such a complex topic with everyone here today, connecting with some folks that I've worked on in this project entirely virtually because of the pandemic. This was one of the only projects where I've never really engaged face-to-face with my colleagues, internal or external, pretty much the whole project. So that was interesting. And today, I'm going to speak to you not so much about the process of risk assessing. That can probably take an entire morning in itself, but really trying to share a couple of key tools that we found useful, specifically in trying to quantitate the risk. So I will go through a little bit of framing the risk assessment process because I think it's very relevant and important when we start to think about how to design and use these tools. But then I will try to share three main tools we found and the industry have found useful. And that will be the value of experimentation and use of model and mean studies to try to inform risk factors in your that could be useful in designing your risk assessments around real products. And then also the development and verification of quantitative models that can be used to help guide and assess what the potential formation, extent, and rate would be in a given product. And then finally, I'd like to talk a little bit about risk with aluminum blister packaging components. This is something that we've been working on since the inception. There's some information out there, and we have a new publication coming out soon. I think that will be insightful into that risk factor, and I'll share some of the data actually for the first time today. First, and this is an approach to assessing risk assessments. And one thing I will make sure I state up front is everything in here is certainly not quantitative. These are snippets of useful or hopefully useful information. And this is just kind of one way to kind of contextualize an approach to assessing the risk. When this all started, and was mentioned the valsartan discovery in 2018 and really started getting rolling for the industry in late 2019, a lot of us scientists really looked in the literature, what's known out there, and we found so little. And this was paralyzing to me joining the team because first question I was like, well, how can I truly assess a risk when I have no data, no literature to work off of? And quickly, industrial consortia started forming and really collaborating on a very high level. It's very impressive how the industry and health authorities work together collaboratively through the whole process. And the other thing that was challenging, but also very exciting to work as a team, is the science was evolving at the same time as the regulatory framework. And that's certainly frustrating, but it's also very exciting as you can collaborate very dynamically. And what the industry was doing, and the partners at Merck and other companies, were trying to leverage as much of those guidances out there. The ICH guidances that were already published are very useful in helping to design a framework. There was nitrosamine specific guidelines coming out, like Article 5.3 from the EMA, that was really driving a lot of how we were trying to frame risk. I mentioned the consortia that we're developing, and also things like the LASA excipient adventure and collaboration that came out. Very, very important work to try to understand risk with excipients that we contributed to as much along with other pharmaceutical companies. And then really trying to look at the risk assessment process, it kind of breaks down in one view as gathering information and then assessing the risk. And those two first lines there, scientific foundation, gathering information. First, what can we learn from literature that's out there with nitrosamines? I mean, they're not a new thing. It's a very powerful chemistry used in synthetic chemistry reaction routes. So we knew something, and we knew stuff about the kinetics, but we just didn't know certain things, and specifically with drug products. That led us to gathering information, experimentation. So when you think about a drug product, there's a lot of valuable information in the dossier, right? You have, things like the P3 document control strategy in your process, P23 with your formulation details and so forth. And also specification limits can be very powerful to try to frame your risk around specific product. But then there's cases where there's risk that's still present. You examine that, and like Andre was saying, you find a secondary mean functionality group in your API, and you're like, okay, there's that. And then we learn more about the excipients, and now there's nitrates. It's like, okay, we're stuck, right? And that's where the experimentation comes in. Can we do some very purposeful experiments using the information framed around that drug product itself, right? They can put constraints and boundary conditions on our experiments and learn something that can help quantitate the risk for that product using conservative assumptions and so forth. And then finally, it's the quantification itself. So we get those experiments, we learn, we get some data, we develop some models, and then we can try to predict formation. And Andre mentioned there's some limits out there, and for these NDSRIs, there's no published literature. So you can use things like the structure-activity relationships to try to estimate what an upper limit would be on a daily basis. And then using your dosage information, you can try to estimate whether we're close to that or not. And if you're close, a lot of times you would unfortunately maybe consider that a risk. But if you're not, you might be able to say, hey, we've done a very thorough assessment and we don't have a risk. So I mentioned gathering information and different factors. So, in doing this, firstly, you have to examine, do you have a vulnerable mean in your process or a source thereof? So do you have a potential functional group in the API, a secondary mean or vulnerable tertiary mean that could be present? Is there potential for carryover of reagents from your chemical synthesis process in your drug substance? Also, it's very important to look at your degradation pathways. So a lot of times pre-formulation reports, even if it's not specified in the dossier, can be very useful, saying, hey, there's some degradation pathways we want to take a deeper look at. And if you identify something there, then you have to say, is there a nitrous aiding source, right? Unfortunately, with what we've learned from excipients, that's often there. And if you happen to say yes for both of those, well, then you're stuck in going a lot deeper in your analysis, right? And that's where looking at the drug substance, drug product, the formulation process is very important. And then, although this is not by far a comprehensive list of factors, what we found is there's a lot of things that are important to consider about your mean. The pKa of that secondary mean group is certainly a very important factor. Solubility and physical form. This is something I'll share some data on. Maybe it wasn't intuitive at the beginning, but we learned things from our model mean studies. And then also the nitrous aiding source. And then for the drug product, we've certainly learned that depending on how you process that drug product, how you store it, what the vapor absorption behavior is of your formulation and your amine, the physical form relationship to that water content, these types of things are very important to consider in your risk assessment. I mentioned the use of a model mean study. So I just want to highlight, there's a publication listed on the bottom that recently came out in Journal of Pharm Sci where it goes into depth on a lot of the experimental studies and what we've learned from this, as well as some hypotheses on how we think nitrosation is occurring in a solid dosage form. So I encourage you to check out for more detail. But I just wanted to kind of share some lessons learned. First of all, we found these type of studies to be very informative. I kind of mentioned already some things we've learned from that. This work really helped to prioritize the risk factors. When you think about nitrosamine formation in a drug product, particularly a solid dosage, you can make a list of 40 factors that could be, I'm just throwing out a number, a large number of factors that could be relevant. And without some science and scientific data, it's hard to do a risk assessment that's comprehensive and that you can stand behind without narrowing that list down. And that's where the model of means have found to be very valuable, where they've worked to confirm the importance of pKa, the water, the form of the amine, whether it's a HCl salt or free base. And also the bottom three things are things that kind of jumped out that maybe weren't so intuitive at the beginning. And that's things like the physical structure, amorphous or crystalline, solubility, or exposure of temperature and humidity during shelf life. And looking at the data on the left-hand side, this is actually for an amine that was processed using a dry blend and compression. The crystallinity of it was a crystalline model amine going into the process. It was verified to be crystalline after the process. And upon storage, we clearly saw at 75% RH, independent of temperature, we saw a large peak in nitrosamine. And we dug in further and we did some vapor absorption on that formulation and on the model amine, and also some variable RH X-ray. And what we found was it picked up moisture very aggressively as the RH relative humidity increased above 60%. And also at the same time at a RH above 65%, from the evidence of the X-ray, it looked like we either lost complete crystallinity to amorphous or certainly a large fraction thereof. So although it can't be comprehensive from that one study, it certainly would suggest that the absorption behavior of this amine and the physical form were very relevant in the spikes we saw in the nitrosamine formation. Another useful approach we found is, and the industry worked very feverishly in that first year in 2020 to culminate all the information that's published out there about the reaction kinetics of in this case, DMA, dimethylamine formation reaction to NDMA. And the authors, and the publications listed at the bottom, the authors put together 5 reaction schemes that are very relevant for a lot of the common pathways that form nitrosamines. And 2 of them are equilibrium reactions, both for dissociation of nitrous acid, as well as either a neutral or protonated state of the amine. And then followed by that, 3 common nitrosation pathways. And they constructed a model, because of the simultaneous nature of these reactions, constructed a model using the Berkeley-Madonna framework. So these equations could be solved simultaneously, and then did some relationships or some predictions using the model to existing published data, and showed that, in fact, the model was doing what it was intended to do. And that was intended to over-predict nitrosamine formation. And the reason the industry wanted to do that is we realized, given the paucity of scientific data and information out there, we wanted to be conservative with our estimates. So if we use the model to predict anything with the appropriate constraints that we put in place based on the product and conservative estimates, or inputs to the model, and we still under-predict the acceptable limit, that it could be a useful tool in helping to frame our risk assessments. And that publication is listed there. That was great, but we also wanted to take another step further to verify the validity of the model. So I stopped short of saying validation, because this is not a validation in the sense that all of us in the room would think about it, but it was a verification that the model was doing what we intended to do. And we took a model amine, 4-phenylpiperidine HCl salt, and looked at and dissolved nitrite in aqueous media, held it for 24 hours at controlled temperature, and then looked at the formation as a function of pH, and then ran the model for those conditions simultaneously. As you can see in the graph, the data there in the circles, the model was over-predicting as it was designed to do, at least certainly at low RH. And another thing is it did show the expected trend of pH, where at low pH around 3, or the pKa of nitrite, we're getting full conversion or close to it for a reactive secondary amine in solution form. Which is very important information. We also ran, it's not shown just from brevity, but we ran a similar experiment at trace nitrite. So numerous products were examined, liquid-based products, to estimate what a nitrite concentration would be based on the excipient data we had at the time. And then we picked 0.1 molar nitrite, ran the simulations, and that same relationship or trend or curve shape as a function of pH was obtained. But we saw very, very little nitrosamine formation, nothing above a 10 ppb level of the analytical method at pH 7 and above, and fairly little even at pH 3. So this is very, very good and important information for us to get to show that the model is doing what we intended to do. Okay, so I have a couple minutes. Is that correct, Andre? All right. I'm going to skip over this. This is, you can read through this. This is just an example of how we can use the model. It's a hypothetical example, but it's a user interface, and then it kind of walks through how you could take information from that you have about the product and then run the model and use the outcome. Finally, I want to talk a little bit about blister packaging and what we've learned there. So the industry, and a lot of this, at least that I've been part of, has been driven by a packaging sub-team in the nitrosamine IQ team to try to understand and dig into more what we know about packaging. Several companies have taken example commercial blister coupons, examined the printing and the overlacquer and the foil to look for presence of nitrocellulose, amines, discussions with the suppliers and so forth. And what we found is that Unfortunately, the inks, the primers, the overlacquers, they can all contain nitrocellulose. And upon heating, nitrocellulose can degrade and form NOx, and this can lead to a very active nitrosating agent. The other thing we found is inadvertently, there could be amines present in the inks that are used to color the blister. So you have a potential theoretical risk there, right? And then we thought about we looked at our common blister processes and what can happen during a blister operation that can cause a nitrosamine to form. And what we found is you're applying pressure and heat to the foil as it seals on top of the blister cavity. So that heat could potentially cause reaction to occur. It could also cause volatilization of the nitrosamines. These temperatures can be in the range of 150 to 250 Celsius. Applied to that heat to get the lacquer or the heat seal lacquer to properly seal. So we took that information and framed it. Okay, so if the heat can cause formation and volatilization, well then how is the nitrosamine going to contaminate the product? Well, that sealing is occurring and its formation, and it seals the aluminum foil barrier to the cavity. So at that point, contamination risk should be minimal or non-existent. But you could volatilize nitrosamines. They could form like a cloud, if you will, or some kind of vapor concentration. And then that could migrate upstream into the open cavities that have your tablets that are just placed in there. And then that could contaminate and you could seal the nitrosamine in there. So we wanted to do an experiment to look at that risk. And we took a commercial sample of a blister foil itself. We characterized it, shown to be a very high concentration compared to some others with nitrocellulose, and had DEA and DMA present. We sealed that in a moisture impervious vial, applied different temperatures to it, and then analyzed the headspace for NDMA and NDA concentration. And what you can see on the left-hand side is we did in fact form volatilized NDMA and NDEA. Significantly more NDMA because it has a higher vapor pressure, presumably, led to a higher rate of formation. Nice thing is we noticed a very linear relationship. So it's a constant rate of formation. If you take that rate, and we took the NDMA rate at the highest temperature to be very conservative, and then you use that rate, you can start to develop a process model to examine what happens to the concentration around your blister line during processing. We used information about what a common air changeover rate would be for a clean room, some examples on what kind of blister production rate we would have, a size of a blister, and then we did a mass balance in that vapor, the vapor around that process. And because of the linear relationship, the constant production rate, and the fact that air is removing nitrosamine also at a constant rate, but that concentration is increasing over time, you will get a plateau, a maximum nitrosamine formation at some point. When that concentration reaches a certain point, you have removal is equal to production, and then it remains constant. You can use that maximum concentration and estimate for this type of example, what's the potential worst case contamination in blister well, and we found that it's about half of a nanogram or 0.5 picograms. So in this example, using this data, we were able to show that for most products in a well-ventilated GMP environment should have minimal risk of contamination during blistering. Now this only speaks to external printed blisters. If you have printing on the inside of a foil that's product contact, that's a different risk scenario. So I mentioned at the beginning, this is only a certain example. So there's other examples that you must take into account. This isn't to say that all blistering is no risk. It's just one example of how to use the data from these experiments in a model to help estimate that. So in conclusion, I just want to say, the collaboration between industry, health authority has been remarkable in my opinion. I've worked on other external consortia, and nothing has compared to the high performing nature of this group in this room and others that I've worked with. We did learn through this work that unfortunately nitrosamines can form in solid drug products. I was joking, I think with Dr. Pauly earlier, that we all went into this thinking this was going to be an easy job in drug product. I certainly did. And it turned out to be nothing but the opposite. We found that models can be very useful. Model mean studies and quantitative models can be useful to assess risk. And also just want to say that there's been a lot of work done in assessing drug products. I think by far the science is still evolving. I think to the point where us product development scientists like to be in developing new products. We're not there yet, right? I think there's more work that we can do. I think hopefully the work that everybody's sharing today can provide a foundation to spur more research so we can really kind of put this to rest going forward. And finally, I just want to acknowledge the various members of the Merck team that supported all this work, the numerous discussions and debates with colleagues internal and external to Merck, a lot of the authors I cited with the publications, really dedicated to making sure we got the science and knowledge out there. And finally, I just want to thank the CRCG team and the FDA for the invitation to speak today and putting together the workshop.
Hello, everyone. So in my presentation today, I'm going to discuss the performance characteristics of the analytical method for liposomal analysis that we observed from inter-laboratory studies that performed together with the other regulatory agencies. How I move it? Yeah, so the data and the results presented in this presentation just represent the view of the authors, but not the agency's policy or views. And the following slides, you already heard from Andre about the drug product quality, Office of Quality, and this just adds some beautiful pictures there. So as mentioned, that the liposomal contamination has continued to be the significant concern of pharmaceutical qualities and safety since 2018, when NDMA was first discovered in blood pressure control medications. And then the realization that the drug substance themselves can also be the origin of the liposomal, leading to the formation of the NDSRI in the drug products, kind of like magnify the scope of the problems. So in this session, you'll hear a lot of presentations discussing about the risk assessment and risk control, and also the strategy developed to mitigate this risk, to address these issues. So overall, this risk control or strategy to mitigate the risk, analytical procedure plays a critical role. So giving the very low acceptable intake limit for many liposomes and the regulatory guidance pertaining those without any carcinogen or genotoxicity data, it is very important to have very sensitive and selective analytical methods for liposomal analysis. So because of this, mass spectrometry is kind of like a preferred or almost defined like a choice of the analytical technique for this usage. And despite being widely used for in many aspects of the pharmaceutical development, the mass spectrometry as a quantitative tool in the quality control of small molecule like pharmaceuticals is still kind of like uncommon and very limited. So the use in the liposomal analysis kind of like raise many questions among the regulators and also the stakeholders that what the performance is like for like a mass spec based liposomal analysis. So how we can evaluate the methods, whether it fits for the purpose. So to further this understanding, we FDA can collaborate with the 5 other international regulatory authorities to perform these interlaboratory studies. So in this study, we wanted to understand the performance through 3 aspects, like accuracy, repeatability, and then reproducibility of these methods. And then we also wanted to have an idea about what the quantitation limit can be, and then the areas of the improvement for the analytical methods. So in this study, we collected some drug substance and products and distributed among the 6 laboratories, participating laboratories, and each laboratories use their own analytical procedures to analyze the small molecule liposomes in these examples and report back to FDA to assemble the results and then analyze the data. So in this presentation, I will briefly provide a very brief like a summary of what we did and what are the key findings from this study. So the first was like a samples. We have two sets of the samples. We have contaminated and then spiked the samples. So the contaminated samples include 3 lots each of the drug substance and the drug products of valsartan and losartan. And these samples contains the liposamine like NDMA, NDEA, or NMBA, like in the various levels. And the use of this analysis of this set of the samples will give us an overall view of the performance of a method from the beginning of the sample preparation to the instrument analysis and then to the data analysis and reporting. And also because of the diversity of the drug products formulations in this set of the samples and also the different manufacturers, analysis of these samples also allow us an opportunity to see whether an analytical method can be used universally to detect liposamines without any modifications. The second set is the spiked samples. So those include 1 lot each of the drug substance, drug products from valsartan, losartan, and those materials did not contain any detectable liposamines. And for this set of samples, the participating laboratories were asked to spike in the 6 liposamine standards at a different level from 0.01 ppm to 1 ppm and analyze reported results. So there are totally like 7 analytical methods used in this study. So that covers like most common of the analytical techniques being used for liposamine analysis, include the LC in combination with high resolution mass spectrometry, and LC in combination with mostly like a triple quad to do the mass analysis, and also the GC mass analysis. So for each laboratories can make like a triplicate samples and analysis for each materials they receive, and then report the results in the concentration of liposamine as a relative to API as a ppm or microgram per gram. And for that, we look at the repeatability, reproducibility, and accuracy of the method with a repeatability that can represent the variation for within like each lab between their sample analysis for one liposamine, and that was represented by the relative standard deviation. And also for the reproducibility, that can reflect the variations between the different methods. So that can also be calculated by the relative standard deviation between the different lab areas and combine all the data reported for one analysis. And accuracy only applied to the spiked samples. So we look at the percentage difference, which is calculated by compare the relative difference between the measured values compared to the spiked values. So we first look at the performance of this method for the spiked samples, and look at the accuracy. So we put together all these percentage difference values as a box plot and organize them by different individual labs. And in each individual lab's color represent the different liposamines. So overall, you can see that the percentage difference can most of them located in between -30 and then to 30. And but there's also definitely a variation, performance variation between different labs and regarding different liposamines. And then we want to look at how the concentration, liposamine types, and sample matrices affect the performance of the accuracy performance. And we put them together as a box plot again. So for the concentration, there's a slightly improvement with the increase of the concentration, but the liposamine types or sample matrices, there's hardly observed a very significant difference between the different variables. And so we move to the repeatability, and again, we put together the percentage RSD values in the box plot and organize them by look at the different lab performance and different type of liposamines. So most of these values, as you can see, are under the 20. And also, again, there's a very lab performance are variable. And we look at the effect of the concentration, nitrosamine types and sample matrices again. And for here that clearly you can see that the concentration can affect the repeatability. At the lower concentration, the variation are slightly higher than the relatively higher concentrations. And then nitrosamine types or matrices hardly have any like obvious difference for the different variables. And moving to the reproducibility, which represents the difference between different lab areas, we see the concentration, the same effect as the repeatability. A lower concentration, we have more variations than the higher concentration. And for the nitrosamine types, we here see that, for example, the NMBA, the distribution is definitely very different for the other nitrosamine types. So this also reflected in previous slides organized by different lab areas. You see the performance of the different labs can vary, and also different nitrosamine performance also vary. So we can summarize the spike sample performance here. So in these tables, we have like a repeatability, like only like less than 2% of them have a value over 20% of the RSD. And for the reproducibility, also a very small portion of them have a higher value over 30. And coming down to the accuracy, we see majority of them kind of like have a very good kind of like a spike recovery. And about like 96% of them can located in the -30 and the 30. So the dependence of the repeatability and the reproducibility on the concentration kind of like agree well with this Hallway's equation, which was first proposed in 1982 to look at to describe the relationship between the concentration and repeatability and the reproducibility. And one of the objectives is also to look at the limit of quantitation that this mass spec-based method can achieve. So based on the previous discussion and the data, we look at the lowest spike level that we did with this study and find that the data not very kind of like different than the higher concentration levels. So we considered that the instrument can easily kind of like comfortably achieve like a 0.01 ppm, like a limit of quantitation, and even can go lower. So now we move to the contaminated samples, and the results reported here is also organized by different labs represented by different colors. And just by look at it, you see the variations between the same samples, kind of like very different results can be reported by different labs. So if we put this into like a repeatability and reproducibility perspective, we see that the repeatability of these contaminated samples are very similar to the spiked samples, while the reproducibility of these samples are very kind of like different than the spike samples. With reproducibility, contaminated samples show a very large variation. So this indicates that each lab, when they use their own method can achieve very consistent results. However, a different method, when they apply to the contaminated samples, they cannot report a very consistent result. So there are many kind of like potential factors contribute to this great variation. So one of the two of them we wanted to point out is probably sample preparation in terms of extraction efficiency and artifact formed during the process. And then the other factor could be also matrix effect. So those things are kind of like hard to identify and characterize in the development of the methods. And we also wanted to emphasize that each analytical method has to be developed fit for purpose and also validated for their unique sample matrices. And there's probably no like universal method can be applied for the general screening of any of these nitrosamine species. So this kind of in conclusion, we think that the trace level of nitrosamine can be measured with accuracy and precision, and a 0.01 ppm limit of quantitation can be achieved or even lower. And we also wanted to, as pointed out, that the importance of the method development and validation. And how this is related to the NDSRI. So our lab has kind of developed multiple NDSRI analytical methods. So overall, we feel like the performance of the NDSRI methods are probably better than the small molecule nitrosamine, likely due to its larger size and for its easy detection by mass spec and quantitation. So with that, I'd like to thank all the participating laboratories for their contribution and each individual contributors. And thank you all for listening to the presentation.
There we go. Thank you. So first of all, a big thank you to the organizing committee, the Center and the FDA for inviting me to speak here today. I'm here to speak about the critical role excipients has to play. The critical role excipients can play in mitigating the formation of nitrosamines without the use of scavengers. Almost there, right? Okay. Yeah, there we go. So in addressing the nitrosamine challenge, we all know market authorization holders are being asked by the regulatory authorities to mitigate the amount of nitrosamine impurities being found or created in drug products. In this context, the FDA and EMA has provided a stepwise approach in assessing the risk, and in that process, excipient manufacturers can support in a couple of steps. Main steps are in providing insight into the level of nitrosamines. This was definitely done at the beginning in 2018, but later also into precursors of the generation of nitrosamines, such as nitrites, that are existing within excipients. Of course, as we go along, then market authorization holders will need to test their levels of nitrosamines in their drug products. And in the case that this is not at the appropriate level, further steps need to be taken. And also here, excipient manufacturers or excipients can be used to mitigate risks further by utilizing the differences between different suppliers, for example, of nitrite levels. And this is a main topic that I'll be addressing today. If we look at the nitrosamine impurities and the different sources, so we were already talking this morning about the small nitrosamines and how they've been addressed, but the key challenges with these NDSRIs. And if we look at the NDSRIs, we've already spoken about a couple of those challenges, but if we break it down, a couple of the risks with regard to meeting the regulatory limits are, one, that these are larger molecular weight molecules. Second of all, the level of amines within the formulation here, the potential of creation of these NDSRIs are higher. And third of all, because these NDSRIs are vast amount of different NDSRIs and not very well quantified in terms of the risk, we have an unknown there, and therefore we've also agreed that as an industry that those limits are extremely low. And fortunately, we do have the ad interim limits set by the EMA or proposed by the EMA, and we'll be utilizing these two limits within our study. So in terms of an introduction of what we're going to be looking today, we're going to be looking at approaches that you can benefit from excipients in reducing nitrosamine formation without the use of scavengers. That can be broken down into three approaches. So first of all, utilizing the difference between suppliers of excipients. You have different average nitrite levels, but you also have different consistencies of these nitrite levels within different suppliers of excipients. You can also utilize the differences between different types of excipients in the type of use. So whether what kind of excipients are you using within your fillers, within your binders, within your disintegrants, and we'll take a look at what potential gains can be gained from looking there. And third of all, also by looking at the processing route, for example, direct compression versus wet granulation. In this presentation, I'll show the scale at which risks can be mitigated, and it will be a model data-driven assessment utilizing the data coming from the publications of Gessel, and also on the levels of different suppliers of excipients and different excipients, and then also the rates of conversions into nitrosamines from a publication from Justin Moser, who was just presenting earlier. I think it's key in the way that we approach this to understand one of the key principles that we took in calculating the risk. And what we did there is we really took the rate limiting reagent to be the nitrites within the excipients rather than the more abundant amines which are in formulation from the API. So very simplified within the model, we really looked at the level of nitrites that are present from the excipients in the total formulation, and then the conversion of those nitrites into nitrosamines. In the model, we took into account a number of other risk factors, such as the potential molecular weight of the NDSRI and the full formulation composition, the type of nitrates, the type of amines present, and therefore the rate of conversion that is there. In the talk will go through two parts. One will show case studies where we really look at the differences, different suppliers of supply of different types of MCC or a different type of lactose can be gained. So what kind of advantages can be gained? And the third case study that we'll show will be adapting the super disintegrant used in formulation to show the advantage that can be gained there. In the final section, we'll show a model where we look at the different variables in a multi-variable space, and then show a risk environment, so to speak, and what excipients could provide in an optimized setting. So going into the first case study where we're looking at an MCC formulation, or a formulation with MCC, and where we're going to be varying that supplier. So what we did is we modeled a directly compressible model formulation where we took the MCC values from the Gessel study. There were 9 suppliers quoted, and we took the average level of nitrites and a worst case and a best case situation where we have the lowest and the highest level of nitrites as per those suppliers quoted. And what we did, we put them into the model formulation where we had a certain amount of API, MCC, lactose, disintegrant, and binders, lubricants, and glidants. The model inputs for these were the molecular weight, the nitrosamine molecular weight was chosen as 400, the tablet weight 200, and the total rate of conversion as per the amine and the manufacturing route was determined at 13%. And what you then see is that if you take that average level of MCC nitrite content as per Gessel and you change the supplier of MCC to the best supplier of MCC, you gain a significant advantage. And if you would be in a situation where you'd have in formulation the MCC with the highest amount of nitrites present, and you would move to the MCC with the lowest nitrites, you'd actually create a decrease of 84% of nitrosamine, theoretical nitrosamine buildup in your formulation. So a really significant step down from where you are. Similarly for lactose, where we modeled the same, but then using a wet granulation formulation. Again, here we took the input from those levels as quoted by Gessel, and there were 8 different lactose suppliers then quoted. And in the model formulation, again, API is there, and then MCC, lactose, disintegrant binder and a lubricant were added. You see that the same model inputs were used, except that we adapted the processing route to wet granulation. And due to that difference, the rate of conversion also goes up. So it goes up from the 13 to the 29%. You again see that if you go from a worst case to a best case scenario, you're really dropping the potential formation of NDSRIs in this model by 80%. A huge step that you can do just by changing the supplier of the excipient to a low nitrite version. Third of all, the impact of super disintegrant type. Here we took that same formulation that we had with in case study 2, the wet granulated version, and all we did here is we started with cross-povidone instead of cross-carmylose. Cross-povidone, as per Butzel, has a significantly higher level of nitrites present, and you see a significant drop just by changing one of the minor components within the formulation of 36%. If you put that all into a table, the different case studies that we saw, case studies 1, 2, and 3, where the reduction goes from 80 to 36% in terms of maximum benefit. You can further optimize the case study 4, where you optimize the cross-carmylose, and you then optimize for both the MCC and the lactose, and you get a further reduction to 89% of the total amount. Go down to DC, and you further decrease the potential nitrosamine impact. From worst case to best case, you're really generating a huge leap only by adapting excipient inputs. In the second stage, we researched the efficacy of excipient optimization in our calculated models, and these were used to illustrate a risk matrix approach on multifactorial inputs. I won't go into the models. You can read them. Below. But we were really looking at the level of excipients before, but we took a look at, let's say, a couple of other variables and therefore risk factors, such as the tablet weight, the molecular weight of the formed NDSRI, and the rate of conversion. We created a matrix to show the risk profile of those different variables. The first matrix that we'll show is the tablet weight, the processing route, and the NDSRI molecular weight, which were varied. And the second matrix will show the amine type and form and the processing route, which are varied. And then we optimize the MCC and lactose suppliers, best and worst case. Graphically, what does that look like if we take a non-optimized versus an optimized? So the red bar, the red line shows a full, let's say, a worst case scenario of lactose and MCC suppliers in formulation, in this formulation, versus the best case of the lowest nitrite containing suppliers of lactose and MCC within the formulation. And what you see over a wide range of tablet weights. So why is tablet weight relevant? Because you simply have more excipient and therefore more NDSRIs that are being formed. And we went from 100 to 1,000 milligrams as a worst case situation. And within this entire field, with an optimized setting of lactose and MCC, you're below that limit of 178, which is the ad interim level for as the EMA has presented. So it gives you some time to study the carcinogenic effect of the NDSRI. In a table, then also varying the weight of the NDSRI between 100, 400, and 1,000. You see that in the optimized setting of MCC and lactose, which is the top line, the top colored line. You see that over a very wide range of tablet weights and molecular weights of the NDSRI, you have a relatively positive setting, whereas in the worst case, you have quite a significant part of the range, which is above the 178 nanograms per day level. You also see that the advantage that you get is more than tenfold, right? Going from best to worst across the range, we're talking about a tenfold increase. So the second matrix that we then said we'd look at is looking at the amine type, the manufacturing route, and the amine form, all resulting in different rates of conversion. Here we clearly see that in this modeled approach that the tertiary amine is a pretty controlled situation. A lot of the modeled scenarios are below that 18 nanogram per day limit. With secondary amines, the situation is a little bit different, and you really want to go to an optimized supplier setup of low nitrite excipients. In summary, excipient selection with low nitrite levels, and with low nitrite levels, what are we talking about at the moment? We're talking about 40 to 100 ppb. We can really significantly help to reduce nitrosamines by up to tenfold compared to a starting formulation. This approach effectively de-risks most formulations down to that 178 nanogram per day limit for NDSRIs. Point to note, of course, is it the Holy Grail? We're not at an 18 nanogram per day level as of yet. So for some cases, yes, additional risk mitigation strategies will be needed. Those will also be discussed today in a little bit more detail. And you can shift from wet granulation to direct compression, but you can also add nitrite scavengers to further decrease to those levels. So with that, I'd again like to thank the audience for their attention, the center and the FDA for having me today, and a special thanks to my colleague Alberto Berardi, who helped me with this presentation and all the studies that he's been performing. Thank you very much.